Hello, my name is Sharma, and in this video I'll be explaining the process of photosynthesis. Here we're zooming in on a chloroplast. Within a plant cell there are things called thylakoids, which are stacked up like quarters, and responsible for photosynthesis. The granite is a stack of these thylakoid discs, each represented by a blue rectangle. Here we have focused on the stroma, which will be discussed later, and the granite, which we're going to zoom in on right now, so we can discuss it more. Here we're inside a thylakoid, which we see the thylakoid membrane shown by the two green lines and a thylakoid space, which is between them. It is in this location where the light-dependent reaction occurs. It is called that as a light source is required to initiate the process, such as the sun. Remember, the whole meaning behind the light reaction is to produce NADPH and ATP, which are two energy sources that initiate the dark reaction, which is the next step of photosynthesis, to ultimately produce sugar. And NADPH starts off as NADP+, and ATP starts off as ADP plus phosphate group. First, the light energy from the sun is going to have sunlight that is going to come in contact with our photosystem 2. And so our photosystem 2 is going to get a whole bunch of pigments, or chlorophyll, to help absorb this light. As the light gets absorbed, the electrons get excited and travel across photosystem 2. They then move towards and across the cytochrome complex. As they do so, the hydrogen ions move their way through the complex. The energy of the electrons passing through pulls them through the complex. This results in a high concentration of hydrogen ions in the thylakoid space. As the electrons move through the electron transport chain, they go through photosystem 1. As they go through it, they charge the NADP plus and the NADPH. As more and more electrons move through it, more and more NADPH gets created, almost like a fully charged battery. Then, water is essential to a plant's survival, as you all know. So there are water molecules that get into the thylakoid space. They then break apart, and two oxygen atoms are released, and oxygen is released to the atmosphere, which humans and animals breathe in. So, for every two water molecules, one oxygen molecule gets created. The remaining hydrogen ions add on to the already great amount of hydrogen ions present in the thylakoid space. Then, this high concentration of hydrogen ions forms a process called chemiosmosis. Chemiosmosis is where hydrogen ions migrate to ATP synthase, and as they do, they pass through the ADP synthase and change the ADP, add another phosphate to it, and turn it to ATP. As a result, a constant flow of NADPH and ATP is formed. It is then this process that triggers our dark reactions, the Calvin cycle, as shown in the next couple slides. The Calvin cycle, as shown here, is the next stage of photosynthesis and takes place in the stroma of the chloroplast. The stage is also known as a light-independent reaction, as the only thing required to start this process is the products of the light-dependent reaction. The first step is carbon fixation, when carbon dioxide and ribulose biphosphate, or RUBP, combine with an enzyme called rubisco to create an unstable 6-carbon molecule with 2 phosphate groups. Here, oxygen is also released. The next stage is reduction. Here, this molecule then splits into 2 molecules of glyceride 3-phosphate. ATP then comes and converts into glyceraldehyde 3-phosphate, and ATP turns to ADP plus a phosphate group. The next stage is that the glyceraldehyde 3-phosphate involves the reduced NADP, which is a result of the light-dependent reaction. This converts it into triose phosphate and releases NADP. So far, 2 ATPs have been used, as well as 2 reduced NADPs, as well as resulted in 2 triose phosphates. However, to make sucrose, the end product of the Calvin cycle requires 6 carbon dioxides and 6 waters to create this. So, the cycle happens 6 times, creating 12 triose phosphate molecules. At this point, 12 ATP and 12 reduced NADP molecules have been used, which all go back to the light-dependent phase. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a fourth G3P molecule, making a 7-carbon chain. Adding to a fifth G3P molecule, a 10-carbon chain is created, now enough to craft 2 more RUBPs. With 3 full RUBPs recreated from the 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains that needs to be had to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. Here, we now have a look at the overall photosynthesis cycle. It is evident that light-dependent and light-independent cycles are interconnected, and the respective products initiate their respective cycles. From a light-dependent cycle, ATP and NADPH initiate the light-independent cycles, and from the light-independent cycle, the ADP and the NADP+ initiate the light-dependent cycle. Now, we'll look at the differences between the 3 types of photosynthesis, C3, C4, and CAM. Most plants, around 85% or so, carry out C3 photosynthesis. As you will see, the name C3 has to do with the type of carbon molecule produced during the process. C3 photosynthesis occurs in the mesophyll cells of the C3 plant, which is located just under the surface of the leaves and other tissues. Let's take a quick look at how photosynthesis occurs in these cells. During C3 photosynthesis, carbon dioxide is taken up and transported directly to the Calvin cycle, where it is fixed to a 3-carbon molecule, thus the name C3. If the stomata is open, CO2 moves in and the oxygen moves out of the cell. But under hot or dry conditions when the stomata are closed, oxygen accumulates, which inhibits the amount of CO3 that can be made. Some plants have evolved an adaptation that allows them to be successful in hot, dry conditions. These plants carry out C4 photosynthesis instead of C3 photosynthesis. During C4 photosynthesis, carbon dioxide moves into the mesophyll cell and is immediately fixed to a 4-carbon molecule, thus the name C4 photosynthesis. But the anatomy of a C4 plant is different from that of a C3 plant. A somewhat simplified version of the process is shown here. In a C4 leaf, chloroplasts are located in mesophyll cells, but they are also located in the bundle sheath cells which surround the leaf vein. Carbon dioxide moves from the mesophyll cells into the bundle sheath cells, where the Calvin cycle occurs. This shields the plant from the oxygen buildup that would otherwise occur when the stomata are closed during dry conditions. A second way of addressing this problem of oxygen is to change the timing of photosynthesis. This is called CAM photosynthesis, and in many ways it is similar to C4 photosynthesis, except the timing of the process changes. During CAM photosynthesis, carbon dioxide is taken up during the night when the stomata are open and is fixed to a 4-carbon molecule. It is then stored until daytime as an intermediate molecule, which gives the CAM cycle its name. This adaptation allows photosynthesis to occur in drier climates than in both C3 and C4 photosynthesis. A great example of a CAM plant is the cactus. Thank you for listening to my video. This is Rishabh Sharma.